I'm curious about how animals enter states of stasis, like torpor, hibernation, or even cryobiosis, and also about what we can learn from these states to eventually advance medicine. I'm Siniša Hrvatin. I'm an assistant professor of biology at MIT, a member of the Whitehead Institute, and a Freeman Hrabowski HHMI scholar.
To start off, can you help to describe what those states are? I mean, I think a lot of people have the idea and they understand maybe the hibernation, but maybe the other two states that you mentioned.
As you say, many people are familiar with the fact that many animals can hibernate, in particular in moderate and cold climates. They spend the whole winter essentially in hibernation. But what people may not be familiar is that hibernation is actually a pretty dynamic state. So rather than animals being continuously in it, they kind of oscillate in and out of these states that we call torpor. So essentially, our body temperature is normally around 37 degrees Celsius. But then, in order to conserve energy over a long period of, let's say, a winter when there's no food around, the animals decrease their metabolism and they lower often their body temperature to very close to ambient temperature, which can be like as cold as 0 degrees Celsius. And anytime they do that, they enter what we call a state of torpor. And then torpor can last a few days, in some animals, actually, even a couple of weeks. And then they kind of come out for a day or so, and then they go back in and out and in and in and out. So they do this for a period of a few months. Each one of those bouts is called a bout of torpor, but the whole duration of this season is called a, hibernation period. So those are the distinctions between those two. And then I mentioned cryobiosis. So this is actually something quite different, but there are animals out there, and the ones that we study are called tardigrades. They're microscopic invertebrates. And those animals can actually survive becoming just frozen completely. So they're not just cold, but they're actually frozen. And they can stay in this frozen state for anywhere between, days to months. We've had them in the laboratory for years, and they can somehow like come out and revive from that state. So this is the spectrum of kind of biology that we love to study.
What kind of animals can go into cryobiosis?
There's certainly invertebrates that can do it. So rotifers, tardigrades, nematodes. So those are kind of tiny, tiny little creatures that live everywhere around. You find them probably in your garden. You find them in forest. You find them up in the Arctic, down in the Antarctic, and anywhere in between. But there are also some larger animals that can do it. So, for instance, there's frogs in the United States. A frog called a wood frog that has been seen being actually also in a frozen state. Its body temperature is maybe closer to like negative 3, 4, 5, 10 degrees Celsius. So not maybe as cold as tardigrades can get, but they can stay in that frozen state as well for a period of time. And then there have been, of course, examples of animals like a Siberian salamander, which have been seen, that are even larger, have been seen frozen in the permafrost. So there is a spectrum of animals. And of course, many are probably aware of the fact that if, now is spring, right? So a lot of life comes out. So there are a lot of insects and other creatures which sometimes in the adult form and sometimes in kind of an embryonic form can survive really cold temperatures. And some of them can also actually survive being frozen. Oftentimes not as successfully, but certainly plenty of eggs, for instance, will then hatch in the spring.
The science fiction, right, of humans also, the idea is can we be frozen and then come back to life at a later date sometimes? So tell me now about your research, and are you looking at it for information for humans, or what are you hoping to look at and find?
I am definitely fascinated about potential human applications. The biology itself is really interesting. So, we are also just purely interested in how do animals survive these states. If we're talking, let's say, warm-blooded animals like humans, we are kind of evolved to function around 37 degrees Celsius. This is our sweet spot. And if you oscillate temperature just a little bit, you get up to 40, 41, 42. You have fevers, and that's pretty dangerous. And if you drop your temperature by a few degrees, that's something we call hypothermia, and that's also pretty dangerous. So we're used to living in a very narrow range. But then you have this whole set of animals that can go way outside that range and whose bodies, whose physiology functions in that range. So we want to understand that because we want to understand in many ways what are the limits of normal mammalian physiology. And we want to look at systems that are kind of pushing these limits to try to find new biology there that perhaps hasn't been discovered before, and new ways that we can apply that to humans. So some of the examples of like things that we, dream could come true. For instance, animals that hibernate, they spend a long period of time without moving their bodies, also a long period of time without consuming food. Normally, this should result in the atrophy of most of the muscles in the body, right? You can imagine if you're in a cast and you're not moving for a long period of time, and also when you're not consuming food, your muscles get used up. But somehow, a lot of these hibernators manage to maintain muscle mass and muscle strength pretty well, despite the fact that they should be losing muscle. So we're trying to, for instance, understand how do they do that? What might be some of the mechanisms behind that? And if we can figure that out, you could imagine how perhaps some of the molecules that are a part of their ways of solving this problem could then be used perhaps in humans to either affect the fact that muscles are lost with age, or that there's a lot of bedridden patients where they lose muscles during chemotherapy and similar treatments. So that's, for instance, one direction where it's not entirely thinking about kind of suspended animation of humans, but something that we think we can learn from these animals and then apply to human medicine.
And one of those is maybe the metabolic rate of humans. Is that something that you have researched? And tell me what you know about that.
Our bodies are, let's say again, at 37 degrees Celsius, and our environment is colder than that. And the reason that that's the case is that our cells produce heat and warm us up to a much higher temperature. This is something that we think is very important for warm-blooded animals, for mammals to live and survive and thrive in the world. But it comes obviously at an expense, and the expense is that you continuously need to heat up the body. So animals that hibernate have mechanisms in them to kind of shut off that heating system and somehow decrease some of this metabolic rate, and along with it lower their body temperature and enter a state that we would call the hypometabolic state. So this hypometabolic state, along with being hypothermic, so cold and low metabolism, appears to have, for instance, some beneficial effects on their lifespan. Animals that hibernate live longer than closely related non-hibernating animals. There's been a study that showed that the amount of time the animal hibernates is kind of additive to its lifespan. So it doesn't age in some sense during hibernation. If anything, it actually protects it a little bit. So like for every day it hibernates, some animals live about 2 days longer. So we think that there might be a combination of the point that you were making, changes in metabolic rate, changes in body temperature, and perhaps some other things that are allowing these animals in these low metabolism and low temperature states to eventually live longer. So this is one way in which we're trying to understand that. I mean, also at the basic level, it's important to understand how metabolic rate is regulated and how much you can change it. And we are learning a lot of this by looking at hibernating animals, because they can really actively change their metabolic rate much more so than we think we humans do.
In locations, let's say geographic locations like in the Northeast of the United States and more north of that, it seems like people get ready for kind of a hibernation psychologically, right? That they are going into the winter months and are going to be hibernating. It's more of a psychological state, I think, than definitely the scientific state of hibernation that you're looking at. But do you ever think about that and maybe a relationship with those intense hibernation biological states?
When we started, we weren't thinking about that quite as much. But as you study hibernation, you realize that it is a seasonal behavior. And let me give you an example of what I think about this. So we study hibernation in this animal called the Syrian hamster. Many people may be familiar with them because they're pets as well. So these animals, normally we keep them in like a 12-hour light, 12-hour dark cycle. So just a normal day. But if you want them to start hibernating, you trick them into thinking that the winter is coming. And the way you do that is you change the light. You make it such that more of the day is dark than light. In our case, 16 hours of the day end up being dark and 8 hours light. And you can also begin to kind of change the temperature of the room down to decrease it. And they receive that cue, and it takes them about 2.5 months, after which they will spontaneously start hibernating. So we don't actually change anything directly in those 2.5 month period, but they somehow, this cue from the environment triggers, hey, winter's coming. I'm going to prepare for the winter. And then they start hibernating, and they hibernate for about 2 to 3 months, and then they spontaneously come out, even though we didn't actually change anything. So this tells us that a lot of these animals have some kind of internal clock where they're preparing for the winter, then they're going through the winter, and they're preparing to come out on the other side. So I think that that is actually a really fundamental clock in the probably brain. It might be in other parts of the body, but let's assume for now that it may be in the brain of animals, because so many animals out there, not only do they live in an environment where there is a day and night, and they have to observe that circadian rhythms, what we call, but also they live in environments where there's seasons, and they anticipate seasons, and they, for instance, forage, and some even will actually, increase food intake in the fall to fatten up for the winter. Then they usually don't reproduce during the winter. They wait for the spring to reproduce. There's all these seasonal cycles. So I do believe that because this is such a core part of mammalian, biology and physiology, that key aspects of that annual clock are actually probably still there in the humans. And that is probably why we, in our own way, are mentally and physiologically, because there are also physiological changes in our bodies that happen between the winter and kind of spring, summer, are preparing for a season. So turns out that actually this clock has not been found in animals yet. So it is an interesting open question in biology. How is the seasonal clock encoded? Like, what keeps time in your brain that it's like November or December or January or, July, August, September?
Let's say for animals that are on more of the equator that don't have those differences in seasons, as well as humans that are living in places along the equator that don't have those changes. Are there parallel differences in their behaviors and their life and their biology?
Yeah, no, it's a fascinating question. And I don't know broadly about the breadth of animals and which ones have and which ones don't have different clocks. But one that I can actually point to is a primate. So as we know, primates were first observed in Africa and in kind of hot tropical climates, and then eventually moved. And one primate that is actually endemic to Madagascar is the lemur. Lemurs, despite the fact that they're generally in a warm environment, will also actually hibernate. But what they care about is the fact that there's dry and wet seasons. And when you're in a long dry season, you may not have access to water, and that's pretty dangerous. So their goal is to survive a long period of drought and lack of water by kind of, entering these different trees and being insulated from the environment, and similarly, actually hibernating. So I don't know what triggers their clock, but there again is a value to having an awareness of where you are in the season.
One other thing that I saw in the release was about how the study of hibernation can also be helpful in terms of organ preservation or neuroprotection. Do you want to talk about that at all?
Typically, when an individual who, let's say, is an organ donor passes away, typically from a tragic accident, um, one would want to make the most out of their precious organs and transplant them into an individual who has a really severe need for them, because maybe their liver is shutting down, maybe their lungs are no longer functioning, maybe their kidneys, and they need that organ for to survive. So one of the processes of that is that you can kind of take an organ and you try to preserve it as best as you can for the transfer between hospitals, or sometimes even across state lines, to go into a hospital where there is a recipient waiting for it. And in that process, you need to do the best you can to preserve an organ. One of the approaches that has been used in the past is to cool the organ down, essentially kind of put it on ice. And the logic was that, well, the metabolic rate is going to decrease, the waste products are going to accumulate less, and it's going to make it. And it is indeed true that that helps compared to just keeping the organ at room temperature. But human organs are not particularly well adapted to surviving such cold temperatures, like being placed on ice, essentially. By contrast, if you look at hibernating animals, you have animals that, voluntarily in a controlled, way, drop their body temperature down to 4 degrees Celsius. This is the temperature of the fridge. And they can stay in there for multiple weeks and then come out. And clearly their organs are just fine in the process of doing that. So the overall question is, can we just learn how hibernators can maintain their organs functioning very well for weeks at a time in a colder environment? And then can we take some of those mechanisms and adaptations and find a way to kind of introduce them into human organs in this process of a transplant? And one of the things that we have done there is, and there's this is a broad question in the field, and there are many scientists working on it. One of the contributions that we have recently made is that we took cells from one of the hibernating organisms and took cells from a non-hibernating, in this case it was human, and did some tests where, like, okay, let's just stick them in the fridge and see how well they survive. And it turns out that the cells from the hibernator survived better than the cells from the non-hibernator. And then we were asking, what about the hibernator cells? Let's them survive in the fridge. And we found at least one important, we call this a pathway, like one important component of the cell that the hibernator has really strongly active. And this is an enzyme that clears out certain reactive oxygen species that are on the membranes. And it turns out that if you take this enzyme, it also exists in human cells, but somehow it's much more effective in the hibernator than in the human cell. And if you increase the activity of this enzyme in the human cell, or if you use just a drug, a small molecule that facilitates the action of the enzyme, but by being its own kind of lipid reducing molecule, then you can actually get the human cells to survive in the cold for much, much longer. So the summary of this is that by looking at a hibernator, we have been able to identify some of the ways that we can actually improve human cell cold tolerance. And we imagine that these drugs that we've identified, one could actually put into tissues and organs as they're being transported between a donor and a recipient, to improve their viability, their function in the cold, so that when they're placed in a new organism, they're more functional than they would otherwise be. And the other one that you alluded to before, I think, is also around the fact that therapeutic hypothermia is a practice in certain cases in medicine. So when you have, let's say, a child that's born and it's been hypoxic for a long period of time, they are in a state that we call hypoxic ischemic encephalopathy, where they're in danger of their brain cells dying from this prolonged period of lack of oxygen. And one of the standards of care is to actually cool down the body a little bit for a couple days. And it turns out that that actually improves the long-term outcomes. It decreases the odds of developmental disabilities and other effects. So this, as well as a few other cases in medicine, essentially there's been a recognition that cooling the body down may have some kind of acute protective state from an injury that is ongoing or has just happened. And there is good evidence that some cells in hibernators are more resistant to this hypoxic injury and similar states. So it would be quite fascinating to try to uncover what are the mechanisms behind that. Some of them may be due to changes in temperature, but some of them may actually not be due to changes in temperature. There may be another layer to it where hibernators are better at surviving these kind of more stressful, colder, low oxygen states. And if we could figure out those mechanisms, that could again improve organ viability in a whole range of situations, from traumatic brain injury, perhaps even stroke, or a heart attack even. So that's I think an important area to explore too.
I mean, it's really fascinating what you're doing. Do you have an idea of what the future looks like in your field?
One of the things that I'm particularly excited about now is that we increasingly have the molecular tools to begin to interrogate these questions at kind of a more mechanistic level. We are helping build some of those tools. For example, we study a lot how the brain controls hibernation, but in the past, there were no ways to access precise populations of neurons in the brain in a hibernating organism. And the brain has like thousands and thousands of types of cells. So you really want to figure out which one of them are relevant and only alter that one. So we're beginning to build tools, and others are as well, that would allow us to manipulate the different circuits in the brain that control hibernation. I think that's going to be a major advance because, again, we'll learn what are some of the ways in which animals actually induce hibernation. So I would say that's one area. Another area that I'm fascinated about is that, a few years ago, our work and work of others have shown that you can actually induce a kind of hibernation-like state, or we also call it a torpor-like state, in animals that don't naturally hibernate. So an animal like a mouse, which a lot of laboratories study. So we've actually been able to induce a kind of long-term torpor state in a mouse that can last for weeks. So this is something that we call, synthetic torpor or torpor-like state. And I think it's a very powerful tool for us to explore because it allows us to put an animal, which is normally at 37 degrees Celsius, at a different body temperature to rewire a lot of its metabolism and to ask, what are the beneficial or deleterious effects of this? What's good about it? What's bad about it? Can we learn something about this state? So kind of part one was new tools to study natural hibernation. Part two is the ability to actually induce hibernation, even in animals that are not hibernating. And we're going to explore this in a lot of different directions ourselves too. And then part three is, I think there's a lot of people interested in these applications. So I'm hoping that we're going to begin to uncover the mechanisms behind the protection from muscle atrophy, the protection of cells from, hypoxia damage, and similar, preservation of cells in the cold, and that eventually will actually put some of those things into the clinic.
Thank you so much for talking to me today. I mean, it's really fascinating. Your work is really incredible to hear more about and to understand.
Curious by Nature is a production of Newswise. More than 7,000 journalists get the Newswise wires. Visit newswise.com and find out how to spread your news today.